one well. What we should observe here are, again, kidney structures. They are a little bit compressed. As far as you can see, the tubules have changed shape because they are compressed by a volume process, which is right next to this field of the normal capillary structures of the kidney. So it starts a little bit here, this type of field which we should observe very strictly. Then it continues here. It's having sort of a fibrous capsule which is demarcating it well from the surrounding structures, or at least it tries to. And here is a huge field of such changes, which is marked with newly formed cells, irregular clusters of cells. Here is a field with fibrosis and partial necrosis at the center. And here what we observe is specifically the most often seen tumor in the kidneys, which is the renal cell clear cell carcinoma. What we should observe at this type of tumor is that on the opposite of normal tumor understandings, where we should expect malignant tumors to be the darker field of the slide, this one is a lighter one. That's because these cells are rich in lipids and glycogen in the cytoplasm, which appears to be optically empty when it's fixed with alcohol. Centrally positioned, we observe a nucleus, and very often we might observe centrally to this nucleus also very pinkish, well-demarcated nucleoli. Depending on the magnification on which we observe nucleoli, we stage this tumor through different Fuhrman grades and stages. But I think that one of you had presentation about Fuhrman staging and grading, Fuhrman scale, which I think is not quite necessarily for your exam. Yet you should mention that by the presence of these nucleoli and by the magnification, you might be able to put this type of tumor through different grading appearances. If we increase it more, you can see that those eosinophilic nucleoli are obvious. This is at the magnification of 400, and we might consider this one as a well-differentiated tumor, or a low grade one. It's gone again. Yeah. That's because I observe it somewhere else where I can move it and share it. Thank you for saying me that it's gone again. As far as you can see, these cells are trying to form something like tubules. They are forming roundish structures, and in between them, there is not much of stroma, which might be either fibrous strands or small fibrotic strands which are suffering hyalinization. The vascular system is well seen, predominant. How the tumor is fed is usually normal for it. Vascular system is more obvious at the capsule of the tumor, which is fibrotic one. And not much of inflammatory reaction observed here, which is marked as stromal reaction around the tumor, and it's mainly predominant by T lymphocytes. CD3, 5, and 8 type, want to know? So, here you will be asked usual stuff about tumors. Grading, staging, what it means, not exactly for this tumor. Also, tissue and cellular atypia, features of cellular atypia, which professor usually asks. Also, what else? Whether the tumor is well demarcated, whether it's infiltrating the capsule. You might say that here are a couple of nests which you observe further, trying to be away from the major tumor field, might be considered as infiltration. But since you don't have a picture of all of the kidney, I think you should not be sure about this one. And pretty much, I think that is all about this type of tumor. Most often seen one, as we mentioned. You should also mention other types of tumors which you know inside of the kidney, either starting from the parenchyma or starting from the urothelium in the renal pelvis. It has disappeared again. Anyway, we finished with this one. It was number 19. Now